Hello dear students, last video I have explained regarding the formation of vesicle which happens with the help of coat protein molecules, that is COP1, COP2 and clathrin coated clathrin coatomers. So in this particular class video I wanted to explain you regarding the fusion of vesicle. So vesicle fusion is also a very important process, and this particular process is very well studied by using synaptic vesicles, because this synaptic vesicle fusion can be easily studied as it is going to be a highly regulated process through the impulse transmission. So therefore people have studied about synaptic vesicle, and the information is found to be the method is and the proteins are similar in all the other vesicles fusion also. So in case of synaptic vesicles, they have found this particular fusion is facilitated by a specific set of proteins called as snare proteins. So these particular snare proteins, they are the fibrillar proteins present in the membrane of the vesicle as well as on the target membrane, and these particular snare proteins which are present on the vesicles, they call them as V snares, and the target membrane is called T snares. These particular snares, they are going to form a complex which is going to facilitate the formation of means fusion of the vesicle. So here these particular proteins, they are also going to be aided by other proteins called snaps, that is these particular yeah, so these snaps are having the receptors, the vesicle associated snap receptors, that is the V snares and T snares. They are the one which will help in the formation fusion of the vesicles, and this fusion of vesicles is going to be plays important role in carrying the target protein molecules or the protein molecules to the specific organelles, or for the secretion into the plasma membrane, or even proteins are going to be targeted into the membrane. So you can see this is the snare complex which is going to happen during the fusion, which will bring the two membranes very close to each other, leading to the fusion. So the T snares, very good example is the SNAP 25, and syntaxin. R snare is the synaptobravin. So I'll give you the detail. So this is, you can see the process. So first the membranes are going to come nearer, the snares forms a complex, and they are going to show a winding through which they are going to come very close with each other. So there it is, synaptic vesicle happening, this is transport vesicle, late endosome, how it is going to meet with the late another late endosome, or vacuole or vesicles, how they are going to combine. So all this happens with the similar methods. So this is an experiment where people have done it in order to measure how exactly the fusion is going to happen. So photolysis of caged calcium chloride, so how it is going to get help in this process, you can see. So initially the vesicles are going to be there at the docking zone, and then they come into active zone, then priming will happen, and then fusion will takes place. So the study of this particular vesicle fusion was pioneered by the scientist called Rothman. So he demonstrated that this particular fusion process can be easily backed by very low concentration of N-ethyl maleimide. So N-ethyl maleimide is a substance which is going to alkylate the cysteine amino acids. So this N-ethyl maleimide, when it was used, they have isolated the proteins which actually shows sensitivity for N-ethyl maleimide or gets inactivated, and they call them as NSF protein molecules. See, these NSF protein molecules have been identified to be cytosolic ATPases, so which will not directly bind with the means which have no role directly with the membranes as a membrane component. but it is found that there is a protein called a SNAP, soluble NSF attachment proteins. So these soluble NSF attachment proteins, they connect the NSF protein molecules to the membrane proteins. So these, because these SNAPs, they have a target receptor on the membrane called snares, that is NSF the SNAP receptors, and these snares, as I mentioned earlier and showed, they are the integral membrane protein molecules, or even they can be lipid linked protein molecules which will be present on the membrane. So these are the how a membrane vesicle membrane is going to be and what kind of proteins it will have. You can see there is cysteine string protein molecule is there, synaptobravin, synaptotagmin, synapsin 1, synaptophysin. Okay, so like that, there is SNV2, there is vesicular transport protein molecules are there, transmitters are there, there are proton pumps are there. So all these are going to be present on the membrane of the vesicle, a typical vesicle. So if we come to the study of these particular vesicle proteins, so these vesicle proteins have found to be they have found to be having similarity in the evolution between the yeast exocytosis neurotransmitter release yeast exocytosis and the neurotransmitter release. Means these proteins are highly conserved and they are been in all these different organisms. So they have studied these particular snares and their importance by using several proteases also. They are all targets for this, like botulinum toxin. So this toxin is going to break these particular snares by proteolytic cleavage, and thereby it affects the vesicle fusion, and specifically which was identified in case of the toxicity of botulinum, which is acting as a neurotoxin because it is going to avoid the vesicle fusion, thereby release of neurotransmitters. Now if you come how exactly this particular vesicles bind and they are going to fuse, as we are discussing about the synaptic vesicle. So here it is a protein called synapsins, is 1A 2B 1A 1B 2A 2B. So these proteins, they are vesicle associated and they actually link the vesicles and vesicles to the cytoskeletal elements, and because of them it is going to be docked at the docking zone in the synapse. So it is found that they are going to be highly regulated through the process of phosphorylation, and that provides a connection between like secondary messengers like protein kinases, target activated protein kinases, and how they are going to vary the vesicles fusion and as well as availability. So people also found that synapsin interactions are also going to get weakened by the phosphorylation, uh, injecting the dephosphoryl related synapsin blocks the neurotransmitter release through CAM kinase. So there is a protein called synaptotagmin, which I was telling you, so which is present on the membrane. So this synaptotagmin, it's a single polypeptide one transmembrane spanning region having a protein, means only one helical domain which is present within that membrane, and a large cytoplasmic domain, and with this particular large cytoplasmic domain it is going to interact with the syntaxin. So this protein kinase C, like homology is seen in case of carboxy terminal of synaptotagmin. So thereby it is going to bind with the calcium. So that means there is two calcium binding sites for this particular in this particular protein, wherein the calcium is going to bind and forms a tetramer with each calcium binding with a cooperativity and consisting of 3 to 4 power of calcium dependence of the release. So in the in vitro studies, people have found that the calcium cooperativity eh, at concentration in the physiological range of 10 to 100 micrometer for the release. So in the absence of calcium, the synaptotagmin will serve as serve as a break for the release, and calcium removes the break and may explain how these particular non-neuronal cells are going to be carrying out. Because see, synaptotagmin, when the concentration of calcium is going to be less at the cell, so it is going to be inactivated by the synaptobravin. So when the calcium concentration increases, this synaptotagmin is going to get activated because the calcium goes and binds to C terminal and relieves it from the inactivated position. So this you can see is the synaptotagmins, which is going to be having the calcium binding sites. Okay, so where with the help of calcium it's going to get regulated. So with the calcium it's going to be the syntaxin. When the calcium goes down, so it is going to be bound with the synaptobravin. So synaptophysin, that is another abundant synaptic vesicle protein molecule, function is not clear. And then synaptobravin, another very important protein, functions as a V snare. So role is the release is the aided by the use of toxins, and it's homologous to the yeast BOS1 and SNC1. It is cleaved the synaptobravin selectively, and when the synaptobravin is cleaved, the release of neurotransmitter is going to get blocked, or otherwise I can say, so the release of the fusion will not take place. Then the soluble protein molecules, that is NSF protein. So these NSF protein molecules are going to hydrolyze the ATP, and thereby they are going to help in the breakdown of snare complex, or in other words they release the snare complex. And the SNAPs, they are going to help in the attachment of NSF proteins. There is NSEC1 or MUNC18 or MUNC13, and syntaxin binding proteins, so likely to bind with the syntaxin when it is not in the snare complex. So this is how the ATP driven snap protein molecules, how what the role they play was the formation of the fusion. So these snare complexes which are going to be wound around each other, so that snare complex is going to get relieved by the activity of the NSF proteins. So now the proteins of the plasma membrane, that's going to be the target membrane. So syntaxin, it's a putative T snare which binds with the N and PRQ type of calcium channels, and SNAP25 and synaptotagmin cleaved by the botulinum toxin. So they found the role is the calcium channels are going to play a very critical role here. So SNAP25 peripheral membrane protein, it binds with the syntaxin and cleaved by again botulinum toxins, and forms a snare complex with the syntaxin and synaptobravin. So how the process of now fusion takes place? The fusion will happen with the three main important steps: docking, priming, fusion. So docking is nothing but wherever the membrane fusion is required, so there the two membranes are going to be brought nearer to each other with a distance of 30 nanometers. So this is called docking. So either a transport vesicle in case of vesicle normal cellular vesicular trafficking, or it can be a what I can say specific synaptic vesicle in case of the vesicle fusion in the synapse. So first the vesicles comes near to it the target, and then they get docked, then followed by the priming will take place, wherein there will be increase in the concentration of calcium and high potassium, elevated calcium and hypertonic sucrose treatment are the elevated concentration of these ions that causes the priming. So where the some of the proteins at the membrane are going to get activated, then the fusion will takes place. The membrane, the vesicles are going to fuse the plasma membrane to release the neurotransmitters. So you can see this is what the overall process will takes place. So first there will be zipping, that is nothing but zipping of the snares. They are also called as Q snares and R snares, because R is arginine, where these snares are going to be rich in arginine, that is the V snares, synaptobravin and VAMP, and in case of Q snares, that is glutamic acid rich, they are T snares, they are syntaxin and SNAP25. So this is how the whole process will happen in the fusion of vesicles. At the there will be formation of zipping, hemifusion, then there will be fusion pore, and then finally completion of fusion will takes place. So this is the whole process. First the docking will takes place, and here very important aspect is that the RAB protein molecule. So these RABs are also going to be very important proteins, because the RAB, they are belongs to the they are the RAB is called as RAS superfamily protein. There are plenty of varieties of RABs are there. So depending upon the particular RAB and its a RAB effector complex, so the specificity in the binding of in the fusion of vesicle is going to takes place. Means, so during the process of fusion of vesicles, it's also need to be taken care that a specific vesicle carrying a particular type of cargo should fuse with the particular organelle on the target membrane. So it should not happen like any membrane can fuse with any particular membrane. So this particular regulation is going to happen with the help of this RAB and RAB effector. As I told you, RAB is the RAS superfamily protein, it is a GTP binding and is having intrinsic GTPase activity also. So first this interaction of activation of RAB will take place, and RAB interacts specifically with its effector protein and forms a complex or a binding. So followed by the interaction of RAB, then there will be assembly of the snares will takes place, or the snares, they are the T snares and R snares, they start winding around each other, and they assemble and form a snare complex. And you can see once the snare complex is done, so then they are going to slowly wound, and the winding of the snares brings the membrane of the vesicle very close to the membrane of the target membrane, and then the formation of fusion pore will take place, and the enlargement of fusion pore, which results in the complete fusion of the vesicles. Once the vesicle fusion is completed, then this particular the complex of the snare will get relieved, because the unwinding is going to take place. The wind snare is called as cis snare complexes, which is going to get relieved to form trans snares by the SNAP and NSF protein, which utilizes ATP and unwinds by rotating. It unwinds the snare complex and relieves the complex, which you can make out at the fourth step here. So it requires ATP, and here you can see how the membrane is going to get fused, and you can see during the fusion whatever the water molecules present between the membranes start moving out as the complex is going to take place, and then finally due to the hydrophobicity of the plasma membrane, the lipids, so these lipids are going to just fuse. The first outer leaflets get fused and the inner leaflet gets fused and forms a pore. Zipping, hemifusion. Hemifusion is a state wherein the outer plasma sorry, leaflet of the vesicle membrane and plasma membrane they fuse, and then the uh, formation of means then the fusion of inner layer will take place, then the fusion pore and enlargement will take place. All these are the same process in means I'm trying to show it in, these are all from different books, They represent the whole process in different ways. In some books you can see nucleation, zipping, and then fusion pore forming, and then budding. They say budding is nothing but again that particular vesicle can come back as a part of the membrane which actually fuse with the target membrane, can again get vesicularized by the process of vesicle formation and can again get come back. So here you can see the role of MUNC18, so which helps in the vesicle fusion, and then synaptotagments. So they are the role of the different RABs. RAB 3, which is ribofilin synaptic vesicle, how the fusion is getting regulated. So as I was telling, RAB is RAS superfamily protein. So 22, 39, and 29 kilodalton protein molecule, GTP binding 1, and there is RAB GF, which is going to be means GF on the surface of the target vesicle, which is the geranyl group on the resulting RAB GTP, and unmasks it and inserts it into the membrane, and then which is going to again bind with the target. Because RAB in general, it will not be more present in the membrane, it's a cytosolic. So activated RAB is going to insert into the target membrane, vesicle membrane. So RAB GTPases, and that tethers the two types of proteins that regulate the vesicle targeting, such as monomeric GTPases, and that's R or ARF family it can be. So different RAB family members are found at each step of vesicle mediated transport. So tethering of proteins such as long fibrous protein molecules, and even tethering the proteins link vesicles to the membrane compartments and compartments to each other. So you can see these are different RABs which are playing a role at different destinations. So it's a RAB cycle, we call it as, how the RAB is going to get activated, inactivated, which facilitates the fusion and docking of the vesicles. So in this picture you can see there are plenty of varieties of RABs at different destinations. For example, at tight junction and at soluble vesicle, secretory vesicles, there will be RAB 3A, B, C, D, RAB 8 at the Golgi complex, secretory Golgi complex, RAB 26, 37. You can see trans-Golgi network, RAB 11 is there. There are plenty of other kind of RABs at Golgi complex also. Between the cisternes of Golgi and endoplasmic reticulum, different RABs. Between early endosome, late endosome, there are different RABs. Lysosome to late endosome to lysosome, another different RAB. So RABs are pretty important proteins. So this is how exactly whole process of fusion takes place in the synaptic vesicle. So from the endosomes, there will be empty vesicle which get filled, forms the synaptic vesicle which goes and docks with then from the docking priming will happen, then priming will result in the formation of fusion 4, then the fusion will complete, and then the fused vesicle for part of the membrane may get again vesicularized through clathrin coated vesicles from early coated vesicle, then early endosome, late endosome, like that structures. So this is how the vesicle mediated transport of proteins will take place between the Golgi complex and the rough endoplasmic reticulum. So with COP1, vesicle forms at the membrane of cis Golgi network. So where the endoplasmic reticulum resident proteins which get vesicularized and will get transported to the rough endoplasmic reticulum, And at the rough endoplasmic reticulum, the fusion of vesicle will take place with the snails, and on the other hand, proteins are going to get synthesized at the membrane of endoplasmic reticulum, and even at lumen, which will get vesicularized by the COP2 and will get uncoated and transported to the cis Golgi network, where the fusion of the this particular membrane portion will be taking place. So these particular vesicles involve in three major types of protein mobilization. First, the secretion of proteins. Second, the transport of proteins in the inside the cell from one organelle to another organelle. They can also involve in the movement of proteins or the molecule from the plasma membrane uh, and through the formation of endosomes. So I'll do one thing. I'll stop here. I'll continue with the experiment of fusion, how they have studied, and then we'll discuss about how this particular method are being employed for the mobilization of protein. What is this protein trafficking? What are the called as signal hypothesis? Everything we'll see. Okay, thank you.